Yeah, so probably we can get started. So thank you very much, everyone, for joining us in this series on generative AI and drug discovery. So I'm Michael Bronstein. I'm professor at Oxford and also chief scientist at Bant AI, who is the sponsor for this series. And it's our great pleasure to have today Brian He from Stanford and the Arc Institute. So he is a professor in the Department of Chemical Engineering and Data Science at Stanford. He got his PhD from MIT and worked in different places, including Google X, Illumina, Salesforce, and so on. And he is also an investigator at the Arc Institute. And today he will be talking about his recent paper called EVO, DNA Foundation Modeling from Molecular to Genome Scale. So I think without further ado, Brian, thanks a lot for joining us, and the floor is yours.
Great, thanks. Thanks, Michael. Let me share my screen. Hopefully you can see this.
Yeah, everything is great.
Okay. All right. So thanks everyone for coming. I'm excited to share some of the work that we've been doing on developing I guess a DNA foundation model that hopefully will be capable of doing sequence modeling and design from molecular to genome to genome scale. So just to begin this presentation, all of you are probably aware that machine learning has kind of revolutionized the study of molecules. For example, AlphaFold is a model that has really almost kind of solved some areas of protein structure prediction, where you can obtain a protein structure with close to atomic level with atomic level resolution that in some cases matches experimental accuracy in its predictions. And also new flavors of AlphaFold and other types of structure prediction let you kind of understand the structure of proteins bound to other proteins or proteins bound to RNA, for example. So molecules are very complicated in and of themselves. So we're definitely still interested in molecules. But when you also consider molecules in the complex of, for example, an entire cell, this cell is several orders of magnitude more complicated than individual molecules. And a cell requires these complex molecular interactions between thousands of molecules working together to form a complete cell and a complete organism or a single cellular organism. So we're interested in definitely understanding the rules of molecules, but also how these molecules interact with the broader kind of organismal and genomic context as well. So when you kind of look at this complexity, it seems quite daunting. It seems quite challenging to model things at this scale. How do we go about doing this? How do we even go about collecting the right training data to get enough information to understand information at this scale? So it turns out that the DNA of an organism actually contains the information that encodes all of the protein and RNA and DNA molecules that make up a complete organism. And actually, people have been sequencing the DNA at whole genome scale and depositing those sequences in public databases. So, in theory, if you could train models on these whole genome DNA sequences, then you could model or get some understanding about the information that goes into creating whole organisms. The problem, though, is that biology is written in a kind of foreign language. So here is a schema of what's called the central dogma of molecular biology, where you have the DNA fundamental code, and that gets transcribed into RNA, and then that RNA gets translated into protein sequences. And at each level of this hierarchy, there's this code that as humans, we don't have really good intuitive we don't have really good intuitive understanding of what these characters or how these characters come together to form these sequences. I've been looking at DNA sequences and protein sequences for a long time now. And if you just gave me a random string of these characters, I probably wouldn't be able to distinguish those from actually real proteins or DNA sequences. So we can't speak this kind of foreign language of DNA, but there are potentially very rich patterns embedded in each of these sequences. So how do we kind of learn the patterns or the statistical distribution of these sequences? We can basically take inspiration from models of sequences that have made tremendous progress in other areas like natural language, for example. And these models, these language models, are trained with quite simple objectives. So, for example, almost all models of say English text right now are trained with this kind of autoregressive or next token prediction objective. So you give it a sequence prefix of words or tokens, so to be or not to, and you predict the next token in the sequence. And we can do the same thing actually for sequences of DNA. So here we have a sequence GTGCATCT. But in this language, I don't have any idea, or I can't predict the next token very intuitively. But to say a model that's learning these patterns from data, the sentence to be or not to be, or the sequence of tokens, this GTGCATCT, it's kind of equally incomprehensible or comprehensible to the model as it's learning these sequence distributions. So why would this be meaningful? Why would we want to learn some of the patterns in say the DNA sequence? So when you think about it, there's the actual DNA sequence, which biologists often refer to as genotype, but there's the rich phenotypes, these protein structures, how these protein molecules and RNA molecules interact with each other to form biological systems, and how those systems come together to form complete organisms. So this is the phenotype layer is actually what we want, and genotype kind of encodes via kind of biological mechanism all of the rich phenotypes that we're interested in. But there's not a lot of data in the phenotype. So there's actually not a lot of, it's very hard, for example, to measure all of the atoms or all of the molecules in a cell. And in biology, a lot of complex phenotypes themselves, there's not great assays for in the laboratory, for example. So we can actually, but because of the rise of next generation sequencing, so our ability to kind of rapidly read the genetic code of these organisms, there's actually a lot of data at the level of genotype. So if we're using kind of large scale machine learning, data-driven methods to understand biology, we kind of want to operate in a very data-rich environment, just kind of by definition. And we can kind of leverage this property that because of evolution, the phenotypes will leave kind of an imprint on the actual genotype or the sequence variation. So evolutionary fitness will be reflected in sequence variation. And when we go out and we sequence these organisms, and we sequence all the protein molecules and all the RNA molecules in these organisms, that distribution of sequences will reflect in some way the evolutionary selection on those organisms that has been going on in nature. And if the evolutionary fitness that leaves its imprint on sequence variation is useful to us in the laboratory, so that the kind of fitness that nature has assigned to these sequences is also somewhat correlated with the fitness that we as humans want to get from these biological sequences, then this is a good kind of prior or good prior information for kind of learning phenotypes or functions and sequences of interest from natural sequence variation. So this is the kind of basic idea of why we want to train on sequences. And actually there's parallels in natural language as well. When we say train language models on data from the internet, that those sequences are not random either. The world leaves kind of imprint on the distribution of sequences from the internet. And so in the same way, the world is leaving its imprint on the sequence distribution of nucleotides as well. So this has led us to this preprint that I'll be discussing in this talk, where we really wanted to take this idea and kind of run with it. And this is a collaboration with Patrick Hsu and Christopher Ray and others at Stanford, at Arc Institute, at Together AI, led by Eric, Michael, and Matt. And this is a model that we're calling EVO, which is a model of genomic sequence from at very long genomic sequence contexts across prokaryotic genomes. So fundamentally, the actual algorithm that the actual training task that we're doing with this model is very simple. We just train a model to predict the next token given some prefix of nucleotide bases. The actual architecture for doing this uses a very large neural network, and actually uses a kind of frontier architecture called striped hyena. So the architecture interleaves kind of rotary attention, or with that we use like a rote position encoding, and kind of modern or like flash attention in these rotary attention layers, interleaved with hyena. And I think there's 10% attention in the architecture. And this model is trained at 7 billion parameters on 300 billion tokens, where each token corresponds to a different DNA base, at 10 to the 22 floating point operations, which is an order of magnitude under the Biden administration's executive order, beyond which we would just have had to report this model to the administration. And so the reason why we're training at this scale, I'll get to in a moment, but scale does somewhat translate into, or scale does translate quite well into improvements in downstream performance, particularly, for example, in the ability to predict the next token. The reason for using this architecture, which I'll also get to in a moment, is kind of twofold. First, these architectures are able to kind of computationally scale to larger sequence lengths. So, we're also training this in kind of an academic nonprofit setting. So efficiency does matter. And we also do care about the sequence lengths that we feed into the model, because we want to be reasoning or synthesizing information at genome scale. But also we think that these architectures help us understand information at byte level tokenization, or single nucleotide tokenization. So importantly to this model, in contrast to say transformers trained on English text, for example, the model that we're using just treats each individual nucleotide as a single token. It's like almost like a bit level model. The context length here is notable because we want to be able to understand information at a single nucleotide resolution and at the level of individual molecules, but we also want to synthesize information of how these molecules interact with a broader kind of genomic context. So just for some context, here are some typical sequence lengths in terms of the nucleotide sequence that goes into encoding for these different molecules throughout biology. So tRNA, a non-coding RNA, or proteins like lysozyme or Cas9. And these models like ESM2 and AlphaFold will operate at the molecular scale. But in contrast, when you think of the genomes of these phages, or even this gene cluster that produces rapamycin, these kind of segments of the genome, or even in some cases complete, or in these cases complete genomes, they're much longer than any individual molecule. So if we have a model that can, for example, operate at longer context lengths, we could, in theory, understand all of the information that goes into, say, a complete phage genome or a very large biosynthetic gene cluster. So in terms of the data that we actually use to train this model, we train it on bacterial and phage genomes. This is work done by, or the data set was actually carefully compiled by Matt Durant, who is a scientist at Arc Institute. And the data set basically consists of these sequence, sequence or species representatives in this database GTDB. So there's actually some amount of kind of redundancy filtering that goes into curating the main sequence data set that we use. And the typical prokaryotic bacterial genome lengths that we're dealing with here are around 3 megabases or 3 million base pairs. So even at 131K context, we can't even fit in the entire genome, but we do train on these raw genome crops. So importantly, we don't feed the model, we just feed the model raw DNA sequence. We don't feed the model any annotation information. It just learns on this raw single nucleotide level, single nucleotide vocabulary, the patterns across these large or long kind of genomic crops. Just to highlight the work that the team put into training these models, this is Michael and Eric and Jeremy and the broader team really was able to engineer pretty reliable large-scale GPU utilization or large-scale distributed pre-training. We were able to get actually pretty good GPU utilization, close to 100%, training the model reliably for multiple weeks at a time without any issues. And the models themselves, so the reason why we need all this compute and we need to train the models at scale is actually this observation originally observed for in natural language, that with a sufficiently large and complex data set, if you just increase the number of parameters or increase the amount of compute that you use to train the model, those increases can translate rather predictably into improvements in model performance in the ability to predict the next token, as measured by this, say, the perplexity that you have in predicting the next token. So we actually, surprisingly or not surprisingly, but we do observe the same scaling laws applied to DNA sequence as well. And actually, we test out different scaling laws for different architectures, and the striped hyena architecture that we use, where we interleave kind of attention and hyena layers, does scale pretty favorably compared to other frontier architectures or more standard transformer architectures. So getting back to why we're training a model on raw DNA sequence to begin with, we wanted to do this not, well, because we do want to learn patterns over DNA, but also if you look at how biology encodes information from the DNA sequence, the same DNA sequence basically encodes all the information needed to, say, encode the protein molecules or non-coding RNA. And people train these kind of task-specific language models on, say, the protein sequence. And if you think about how say, a protein language model is trained, so I've worked on training protein language models myself. If you look at the training data set of, say, a protein language model, the way people construct the training data set is they'll actually sequence the genomes from organisms in nature. And many of the protein sequence databases, like UniProt, for example, they just take the genome of an organism that's been sequenced, and then they'll just subset or just cut out the sections of the genome that code for the protein sequence, and then translate those using the genetic code from the nucleic acid sequence to the protein sequence. So a protein language model is almost like a very special case of a genome language model, where you cut out the proteins from their genomic context and you only train on the protein sequence. Similar things, you do something similar for RNA language models, where you cut out the segments of the genome that encode for non-coding RNAs and train only on those segments of the genome. In contrast, if you did have access to the whole genome sequences, which we actually do in a lot of cases in many organisms, particularly in these prokaryotic organisms, then you can train definitely on the protein coding sequences and the non-coding RNA sequences, but you can also train on the information from these proteins within the broader genomic context that these proteins kind of exist in. So in a sense, some people kind of think of Evo and this model as kind of a DNA model, but kind of the reason why that I was interested in working on this problem is not so much the actual DNA parts of the model, well, but those parts are interesting as well. But because if you train a DNA language model, then you also get information about all the proteins and all the non-coding RNAs and these molecules that make up a genome sequence. So in theory, and as we'll see in practice, a genome language model is both, or is it's a DNA language model, definitely, but it's also a protein language model, and it can also be a non-coding RNA language model as well. So here are some preliminary results where we take these deep mutational scanning experiments. So this is where someone has made in the laboratory many mutations, say to a protein sequence, and then using some high throughput technology, measured some definition of fitness, say the fluorescence of a protein or a protein's stability in the laboratory for each of these mutations. And for proteins, people have kind of realized that the likelihoods that, say, a protein language model assigns to a protein sequence actually correlates in a zero-shot fashion with the experimental fitness values from these deep mutational scanning experiments without any kind of supervision, without giving the protein language model any information about, or any explicit information about protein fitness or the fitness definition of interest. So this has led to a way of benchmarking these protein language models. So we actually use this protein gym benchmark from Debbie Marks's group. And we can also use this to benchmark the zero-shot function prediction performance of a nucleotide language model that's trained on protein sequences, where instead of feeding in the protein sequence into the model, we just feed in the corresponding nucleotide sequence with the actual nucleotide changes made in those original DMS studies. But in principle, people can also do this for non-coding RNA. You can make many changes to a non-coding RNA sequence and measure some definition of fitness. And in this study, they actually make different combinations of promoter ribosome binding site sequences. And depending on these combinations, they can influence the mRNA or protein expression of a downstream reporter gene. So different combinations actually have different effects on expression. So we can see across all these tasks, the Evo model actually has comparable, some in a couple cases actually better performance than models trained only on the crops of protein sequences or specialized to RNA sequences. So for example, in the protein kind of benchmark, and this is for E. coli proteins, Evo is better than a model, the GenSlim, which is trained on the coding sequences of genomes. And it's comparable, surprisingly, to models of protein sequences that are specialized to protein sequences. Same for RNA, actually. So Evo actually does better at predicting the, in a zero-shot way. It does better than predicting the effects of mutations on non-coding RNA fitness than RNA-FM, which is an RNA-specific model. And these other nucleotide models actually don't even train, or many of them don't even train on the regulatory sequence. So we're at least able to outperform these models in this regulatory task. We do focus mostly on zero-shot performance for benchmarking Evo, because we wanted to kind of establish Evo as a really good foundation layer for these different tasks. So without any additional fine-tuning or attempt to address this kind of foundational layer of modeling, we go head to head with those models, and we're competitive, if not in some cases better. And I should emphasize that EVO is only trained on raw genome crops. So we don't actually give it, say for a protein sequence, any information, any explicit coding sequence annotation. It needs to learn what a start codon is, what a stop codon is directly from data, and it actually does do things like that. Something that's really intriguing, though, and quite tantalizing in my opinion, is that EVO can also begin to place gene or protein function within a broader genomic context. So let me explain the experiment that we did to kind of probe what EVO is learning. So we can basically take a very long genome segment up to 66 kb, and for some genomes this is longer than the entire genome segment. So in some cases we even feed the entire genome sequence into EVO. And what we'll do is we'll insert a premature stop codon at each coding sequence in the genome. So we're basically making a 3 nucleotide insertion, and or a small number, maybe like 12 nucleotide insertion into, say, a 66 kb sequence or an 8 kb sequence for each coding sequence in the genome. And then we compute the likelihood of the mutated sequence, and we compute the likelihood ratio of that mutated sequence with respect to the likelihood of the wild type sequence. And what we find when we do this is when we make this premature stop codon insertion into an essential gene, those likelihoods are much more, or the change in likelihoods, the change in log likelihoods, I guess, of the mutated sequence with respect to the wild type sequence is much more negative than when we make this premature stop codon insertion into a non-essential gene. And so in this way, we can actually kind of do basically zero-shot gene essentiality prediction, or at the very least, the model is able to understand which genes are essential based on like a perturbational procedure that's very close to how you would actually do this study in the laboratory. So, for example, this study from P et al. In lambda-phage, they did a genome-wide CRISPR-I, so a genome-wide CRISPR-I perturbation study on all the genes in lambda-phage. And when you look at this, the effects that EVO is learning over these genomes in this kind of premature stop codon analysis, we actually predict quite well, just in this kind of zero-shot way, which genes were essential, and somewhat quite actually predictive of the actual experimental results from just a CRISPR-I study. So, and the reason why I think this was kind of surprising to me and actually quite striking is not so much the gene essentiality kind of method component, but actually kind of the way that we achieve this. So this is showing that EVO and kind of the density model learned by EVO is maybe approximating is approximating or simulating kind of how this would actually work in nature, and how this kind of genotype to phenotype relationship would occur in nature. So we just mutate the sequence, we genetically perturb it, we don't provide the model with any information, any other information about what an essential gene is. So the model, when you think about it, it has to know where the coding sequences are, at least, or that the mutation is in some kind of coding sequence, and that coding sequence is at the minimum conserved, or is included in a lot of genomes and is therefore essential, and it needs to be sensitive to these single nucleotide changes. So a lot needs to go on inside this model in order to accomplish this kind of performance. So we're interested in using EVO for these predictive tasks, but we can also, because this model is an autoregressive model, we can also apply it to generative tasks as well. So in particular, we wanted to apply this model to generative tasks that take advantage of this the multi-gene nature of the model. So for example, a generative model at the level of individual protein molecules or protein complexes, you can maybe understand things at the level of your 1 or 2 protein chains. But when you add RNA into the mix, for example, then things get a lot more complicated. But because Evo is kind of natively multimodal across these modalities within the central dogma, we can design these multi-gene systems that include interactions between protein and RNA quite naturally. So, for example, Cas, the CRISPR-Cas systems are kind of the main kind of molecules that mediate CRISPR-Cas gene editing is this very large protein, this very large Cas protein. So Cas9, for example, is 1,300, 1,400 amino acids bound to a guide RNA that's sometimes a single RNA segment, or there's an ancillary kind of tracer RNA for Cas9. And so this very complicated complex gets kind of formed together in order to mediate CRISPR-Cas gene editing. And I should say that also the Cas protein at 1,400 amino acids is longer even than the context lengths of most, if not all, kind of protein language models, which typically train at 1024 context. So we took Evo, we fine-tuned it on CRISPR-Cas sequences from different kind of subtypes of Cas, and appended special tokens to be able to prompt for these different Cas subtypes. And because the RNAs, the non-coding RNAs and the protein for Cas are located together on the same operon, we can actually train the model to learn kind of the joint distribution over the Cas proteins and the associated RNAs. I should say that actually designing things at 8 kb length autoregressive model and getting the sampling right is actually quite tricky and difficult. So even getting coherent sampling and samples that kind of look and feel like Cas9 is actually quite non-trivial. But we were actually able to, using AlphaFold as kind of an oracle, actually get sequences that had predicted AlphaFold structures that resemble Cas9 sequences. And so this is what's being shown here, these AlphaFold predicted structures kind of docked with the a guide RNA, though the model does co-design the Cas protein and the corresponding guide on the sequences. Another example is that another example of this kind of design is this IS605 mobile genetic element, which encodes 3 genes. Actually, there's some gene sequences that actually share some DNA, and sometimes the TMPA and sometimes these 2 genes actually overlap as well. So there's kind of 3 genes that make up this system. There's a TMPA transposase and the TMPB that binds to an omega RNA. The TMPA and the TMPB actually don't physically bind with each other, but they do co-occur with each other in many mobile genetic elements. And some people do hypothesize that maybe there are some interactions between TMPA and TMPB that, if you ask some people, they might think that actually these 2 do somewhat interact or maybe share information. But that's actually an open question, so we don't know for sure. But in any case, Evo can actually co-design all 3 of these genes together. So, for example, this is an example Evo generated IS605 sequence with actually overlaps between these 3 genes, and the corresponding ESM fold predictions have pretty high confidence, but the sequences have low sequence identity to things in the training data or to well-established IS605s. And we can also design a 208 base pair omega RNA with interesting and what we think is somewhat biologically meaningful secondary structure predictions as well. A final thing we did, which is kind of a little bit crazy, is that we wanted to test Evo in its ability to design large or long DNA sequences. Basically, we just unconditionally sample, or we prompt with a very simple species level token, and we just let the model run and sample on a single GPU, actually, 650 base pair long segments. And when we do this, we actually get genomes that at a very high level begin to resemble the kind of architecture that we see in known prokaryotic genomes. So there's actually this tool called CheckM developed originally to assess genomes that you would sequence from nature. So someone goes and sequences deep sea vents, or they sequence dirt, or they sequence sewage, and you assemble a large microbial contig, or a large DNA sequence from nature. And a big question is this genome that you assembled even a real genome? Is it accurate? Is it reflective of actual biology? And we kind of repurpose this tool for assessing the quality of natural sequences, but we repurpose it to assess the quality of AI-generated sequences as well. So a lot of metrics that we're using here are kind of borrowed from CheckM. One big metric, for example, is predicted coding density. So CheckM uses a program called Prodigal to predict the open reading frames on a genome. And when we do this, when we look at this, the coding density of Evo-generated genomes actually does almost look like the coding density seen in natural genomes. Another thing that you see in natural genomes is kind of this operon-like cluster where you have genes on both strands, and genes that are on the same strand often will be, or genes that are proximal will actually often be on the same strand. And we also see this kind of operon-like structure in the coding sequences of these genomes. And actually, very interestingly, when we predict the structures of these open reading frames, or the corresponding amino acid sequence from these open reading frames, they do have some predicted secondary structure. Some of these predicted structural motifs do, under a large fold-seq search, do map to proteins in nature. But I should say, and of course, this level of scale is again several orders of magnitude larger than, say, even designing the Cas protein. So this is quite a large scale of design. And I should say that these genomes, we actually don't think that if you put them into an organism right now, they'll lead to a replication competent minimal genome. They're missing key things. Like, I think we do detect some tRNAs. I think we have 18 unique codons that we cover in these genome sequences, but they're missing key marker genes like ribosomal RNAs, or very highly conserved genes that are common across E. coli, or common across prokaryotes. And so, but at a high level, these sequences do sort of resemble natural genomes. And a close analog is, if you did unconditional sampling out of GPT-4 for very long lengths, you might get kind of a natural language equivalent of what we're seeing here. Cool. So in summary, we're training this genome scale foundation model, and we really do kind of see this as an effort to build a foundation model for biology. And we do use this term kind of deliberately, in the sense that, for example, if you train a really good RNA language model, or a really good protein language model, a really good protein language model will not tell you anything about RNA molecules, for example. But if we do a really good job developing a long context genome foundation model, then we could potentially help out with tasks across the entire central dogma of molecular biology. We could help out with tasks in DNA sequence itself, certainly, but also tasks in protein design and prediction, in RNA design and prediction. And we can also help out in tasks ranging from molecular to genome scale. So the theoretical impact of training a model like this is actually quite large. We're also aware that training these models does potentially bring up some concerns about dual use and the ability to design more complicated systems that can be used for good or nefarious purposes. So we are working with really wonderful collaborators at Stanford who are expert bioethicists in developing a framework for the safe and transparent evaluation of these models. We describe all of these modeling details. I omitted a lot of information for the sake of time, but if you're interested, you can take a look at our preprint. We also make the model weights and the code for running the model completely open source. So I just direct you to this kind of URL where you can kind of see, or you can take a look at the models and take them for a spin. Yes. And finally, but last but not least, this is really, really interdisciplinary work that spanned a number of institutions and a lot of very talented co-authors and collaborators. So I just wanted to highlight them here. I think Eric really helped drive this project from in all aspects, and Patrick also from the biology, the expertise in molecular tools development in genome programming and design. Matt was our kind of expert prokaryotic genome wizard and our mobile genetic element wizard as well. Michael was our the core of the ML engineering and scaling expertise, and in the architecture development and science of architecture development and deep learning as well, and a number of other folks on the team as well, contributing to various aspects of the machine learning and the biology. So I wanted to just explicitly call them out here. And with that, that concludes everything that I had prepared. And so thank you all for your attention, and I'm happy to take questions.
Thank you very much, Brian. I think very clear and very great presentation. So there is actually quite a few questions, and we have time for, I think, taking a few of them. So, well, let me maybe start from the most recent ones. Thank you so much for sharing cool insights. Can Evo also predict effects of single mutation and epistatic effect of mutations on coding regions of prokaryotes?
Yeah. So we only look at well, actually, I think some of these data sets. Let me jump back to the slide. So these data sets are at a single nucleotide or single amino acid mutation level in terms of the sequence distributions of these sequences. But also some of these do have combinations. We actually don't explicitly look for our ability to predict kind of nonlinear interactions between the mutations. But yes, I would say we can actually have competitive performance at predicting the effects of single or multiple mutations in the coding sequences.
Thank you. And then, well, similar questions. The focus on prokaryotes is due to simple regulatory mechanisms and extra exon-intron splicing. Would you think diverse eukaryotic sequences would help as well for general foundational model, e.g. folding? And then also, how applicable do you think the current model is for projects in human cells or eukaryotes in general?
Yeah, I think the current model is not very helpful for humans, though maybe for sequences with very close bacterial homologs or something. But we do analysis in the supplement of the paper where we do the same analysis but for human proteins, and for most of those proteins the correlation is actually quite low because the model has not understood the human proteins. But actually there's one human protein where it actually maybe seems to understand it okay. Maybe because that protein has bacterial homologs, or it's also transferring information at a kind of a semantic amino acid level too. We do think though that the ideas in this paper do naturally apply to eukaryotic genomes, or at the very least certain segments of eukaryotic genomes. And I think you, the person who asked this question has some intuition about where the parts of the eukaryotic genomes where this model could be helpful. So yeah, definitely in encoding sequences or in gene islands, I think they're the same ideas potentially work well.
Thank you. And another question, have you looked at codon efficiency optimization tasks as well? One of the big advantages for protein tasks is obviously that you would be able to pick up codon differences for the same amino acids.
Yeah, we haven't looked at this actually, but these deep mutational scan data sets that we benchmark on actually they have codon level and nucleotide level information. So we would maybe be able to tease apart those differences. We haven't looked at it though. But I can't imagine either the model zero shot or a model fine-tuned on top of the model to actually do codon optimization would be within the scope of this effort. Yeah. Good
So
question.
another question, well, a lot of questions. So just stop me when you think that.
No, these are good questions.
Well, which is obviously evidence, so that the talk is good. When you measure a change in fitness, is there any way to predict higher fitness than the wild type? For example, in cases where the protein isn't fully optimized, or are you taking the wild type as the highest fitness?
Yeah, I think in many of these data sets, actually the wild type is close to the fitness peak. And what happens in these deep mutational scanning data sets is that many of the mutations are just lower fitness in the wild type. There are maybe some that are a handful that are higher fitness in the wild type. What this benchmark, this protein gym benchmark and things like it, they're mostly assessing the kind of lower than wild type part of the fitness landscape. But actually, from other work that I've done and others have done, is for protein language models, if you look at the mutations that have higher protein language model likelihood than wild type, and you just use those variants and you construct a library of those variants and test them in the laboratory, they are substantially enriched for variants that improve fitness over the wild type, especially if your wild type variant is kind of not near a fitness peak. So if there is a lot of room to grow, or if there's a space of mutations that improve fitness. And the reason why we think this works is these language models, kind of as shown by this protein gym benchmark, are really good at identifying this clearly unproductive space of mutations. So you remove these clearly bad mutations, you kind of indirectly enrich for mutations that improve fitness. So this is work that's been done in protein language modeling. I would not see any reason why the same ideas would not also apply to language modeling at the nucleotide layer of information. And what's actually pretty interesting, pretty cool for this is if you could expand beyond mutations to the coding sequence, you could actually reach into the non-coding sequence, for example, to improve fitness. For definitely in bacteria, but also maybe in human protein, in eukaryotic sequences as well. This could be a case. So anyway, I think that the short answer to the question is this benchmark mainly tests things that are less than wild type, but language models can help you guide evolution beyond the wild type sequence in terms of fitness. And I would not see why these genome language models couldn't do the same thing, but just even more powerful because they understand kind of the non-coding sequence. They understand codon usage as well.
Thank you. Well, another question, do you have any needle in the haystack experiments that show if you have equal performance across the whole context for example, in the stop codon essential gene experiments?
Yeah, it's a good point. So these coding sequences are actually distributed throughout the genome sequence. So the way that these coding sequences work, or the way that prokaryotic genomes work, is that they're very coding rich. So that the coding sequences are just lined up one next to each other. And we have a separate kind of control showing that like the position of the genes themselves are not predictive of essentiality. So implicit in this kind of analysis is that the actual mutation, these premature stop codons are inserted at different positions into the genome. We haven't bucketed this actually we haven't bucketed this into kind of the ranges, you know, or the percentiles or the deciles or quartiles or whatever of the genome. That would be interesting to do actually. But yeah, so it's a good question. I think a modification of this where we show that this is not sensitive, so it's not like only predicting essential genes at the beginning or at the end of the genome. That could be interesting to show.
Thank you. And can you say a bit more about how you condition sampling to get evo to output molecules with certain functions?
Yeah, so it's the way we're doing it right now is pretty simple. So we have a base model, for example, for Cas. So we have our base model that's just trained on raw genome sequences, nucleotide sequences. And then we actually do metagenomic mining or genome mining. So we actually query a data, we query existing sequence data sets from natural genomes for examples of say CRISPR-Cas kind of operons. And then we can also label, we can see what Cas protein is in this sequence, whether it's a Cas9 or Cas12 or Cas13. So actually prepend a special prompt that just a single token prompt that actually indicates the subtype of the Cas protein. And then we just fine-tune the model. We fine-tune the entire model on this subset of CRISPR-Cas sequences. And then in terms of actually conditioning the model during sampling, we just condition the model with the actual special prompting token and sample from that. That's what we did for this for these results in the paper.
Thank you. And could you also elaborate on redundancy removal or clustering in the training set, and have a look at data filtering and found any relation to performance here? And one example is that we know that for AlphaFold, about 10 or 15% of the data is sufficient for nearly full performance. If we have full fold diversity.
Yeah, we actually really did want to be like very careful about the composition of the training data set. There's these results from AlphaFold that are alluded to that where I also when training protein language models like ESM, there's actually a lot of work that goes into, or there's been a lot of work showing that data set quality and composition and redundancy matters a lot. So we also for this wanted to do the same thing. We don't have a lot of head-to-head experiments where we train on non-redundant data, we train on redundant data, but keeping everything fixed just because we're compute bound. So we just opted for the best practices borrowed from training, say, protein language models, but using those best practices, that is true. So using those best practices is why we kind of opted for the training data set composition that we did, which is a somewhat species non-redundant data set. So GTDB is this data set of prokaryotic genomes, and they do some non-redundant filtering. So they do have these species representatives and some score. I think it's called ANI or something. Some score that filters out or makes sure that the database is like somewhat non-redundant, and we only train on those representatives. So that is yeah, I don't have data on if we had just trained on a redundant version of GTB, the performance would degrade. Though I think those results are pretty clear in other biological sequence models. So we just use those results.
Thank you. And question from John, why do you think the ribosomal genes are missing?
Yeah, I don't know. So I think this is alluding to this result in this kind of unconditional sampling run where we look for, tRNAs and rRNAs for check-in, and we actually do find tRNAs, but no rRNAs. I don't know. It's because there are those RNAs are quite conserved, and certain rRNAs are not long either. Like the 5S rRNA is quite short, so that the model should have picked it up. I'm not sure. Yeah, I think further iterations of this, we will, rather than just completely naive unconditional sampling of the model, could definitely try to align the model or kind of bias the model towards samples that do have all of the ribosomal components. So yeah, but it was an interesting observation. We just observed it and we reported it and I don't know why. Yeah.
So you're saying no particular reason, or at least it's not known.
If anything, yeah, I would suspect there to be some RNAs there. If anything, this just indicates the model and the way we sample from it, just really unconditional sampling. It's not, if you put this genome and this genome alone into an organism, it's not going to start self-replicating, for example. But other statistics, though, that we do compute over these genomes do look kind of interesting. So we have this kind of analogy to the early days of image generation, where you get these images that look very blurry, but in terms of the high level architecture of the images, you can see that things were in the right place. You had like eyes and a nose and a mouth, but everything was quite blurry. So in here we have the operon-like structure. We have coding sequences. The start-stop codons are there. Some of the nucleotide distributions within the genes also mirror natural genomes, which we didn't report in the preprint, but we observed later on. And so at a high level, the genomes kind of look like a blurry picture of a natural genome, but in these kind of key details, like getting complete tRNA repertoires or getting all the ribosomal genes, they do miss those at the moment.
Well, and maybe I know that we are at time, so maybe last question. Did you actually test any of the editors, the CAS in your examples, in the wet lab? And
Yeah, we are in the process of validating some of these designs. Yeah.
does it really work?
We'll see. Yeah, we'll see.
Well, Brian, thanks a lot. Thanks everybody as well for joining, and thank you very much. Have a good day.
Cool. Thanks, everyone. Thanks, Michael.